Well, thank you all for attending. I'm delighted to be here. As we were hearing, I'm from South Georgia, Columbus, Georgia, and because I will probably stay in my slow Southern speech patterns, I would love to have your questions during this, so raise your hands. It's just fine. If a lot of it happens, I probably will get stuck on this slide given the pace of my talk. But please do feel free. If there's something that's not explained well or you want to follow up on a topic, I'm happy to. So I was invited to talk to you about the research that we do and put it in a broader context. So what I'm going to tell you about are some exciting new studies we've pursued related to autism and animal models of autism. And you might be questioning, you know, in your head, what does an autistic mouse look like? And I'll try to describe some of the features of what we think is a good model for this disorder that might help us with the development of novel therapeutics. I am a professor in the Department of Pharmacology, so this is a keen interest of mine. But I also want to be fair to critics of the use of animal models to model something as complicated as a neuropsychiatric disorder. Our brains are bigger and much more complex than the animals we're studying, but we can learn a lot. And I want to try to take you through some of that. I believe last week, those of you may have attended Dr. Emerson's lectures, and he talked about how we can study animal models and learn from them, from their behavior and their physiology. And I'll expand upon that a bit. And I think it was, for those of you who did come, that would be a very good setup for my lectures. So what I will describe, get my pointer correct, is the outline here for the presentation will focus primarily on one molecule, and I'm sorry that if I can't turn around and point to all the screens, but anyway, well, I'm just going to stay with the center one. And so we'll be focusing on serotonin and a protein called the serotonin transporter protein, which I'll describe to you in a few minutes. And I want to tell you about the roles that serotonin plays in the brain and a novel role that it plays in brain development that we're just learning about. I then want to connect for you why we would study serotonin in autism when you probably have heard quite a bit about serotonin associated with other things like depression. But I'll try to convince you that there's something to study in autism as well. And then lastly, I want to bring to you a model that involves manipulating serotonin genetically and what we are learning about mechanisms that may support this very complicated but increasingly commonly diagnosed disorder. So when we talk about animal models of a disorder, I'm reminded of a quote from Howard Skipper, who really introduced the use of mouse models in the study of cancer. And it's been an extraordinary, of extraordinary benefit to cancer researchers to be able to induce tumor formation and treat those tumors and learn about their progression, metastasis, using animal models. And it's amazing to think, not long ago, all cancer biology was studied in a dish, and there are many limitations to that work. And of course, if we're going to try to understand something as complex as human behavior, studying cells and culture, which we do, it can't be an end to the research plan. So Howard said, a model is a lie that helps you see the truth. And what he was trying to say there, I think, is that we can only get so close to what we're trying to model. And the higher, more complex the process, the further away we have to realize we're going to be from the truth. But it will help us glimpse it. We can see it in the distance, we can test some ideas, and we can ultimately take those ideas into therapy for man. But we're also very aware of the limitations of those models. So why study animals and use animal models in biology at all? Well, for me, there's three major reasons. One is that under the study of the normal range of behavior in the animal kingdom is fascinating. Many of us have pets, and we know how different behavior is in different, let's say, dog breeds. And I have a Sheltie that is as hyper as they come. And in fact, I sent my wife a link this morning to a new class of medications that I hope will help this dog, which my wife, of course, attributes to my lack of engagement with him, sort of the frozen father syndrome, and that I don't have enough time to exercise him. And, you know, we have to meet somewhere in the middle, but I think he's somewhere out there in terms of his range of behavior, and it's fascinating. And I have friends who have golden retrievers that just kind of flop down and are just quiet and nice. And, you know, what is it about the brains of these animals that gives them their different behavioral attributes, and how has that evolved over time? How has it evolved through selective breeding? That's fascinating. Brains are wired differently in these animals, and they use their experience differently. We're a long way from really understanding how that is. The brain's the most complicated piece of matter in the universe. And so, but those of us who are interested in brain disorders often are really just interested in how the brain is put together and works. So I'm often reminded that when we have our public outreach lectures from the medical school, we tend to focus on a disease X, Y, or Z, thinking that that's all the public cares about. I think that's wrong. I think there's a lot of people, and particularly young people, that get fascinated by science, by thinking about just how we dream, what makes us laugh at a biological level. So secondly, changes in brain structure and function do have an effect, and they have an effect on behavior whether you're a cricket or a mouse or a human. There are things we can learn at a very fine level by studying brain structure and function in ways that we are just not going to be able to do for some time, I think, in the study of the human brain. So we can get as close as we can and hopefully learn some important lessons. And then lastly, of course, where my profession comes in is developing new molecular therapeutics to try and treat and improve the lives of people with disabilities. Now, I wanted to start by giving you an example of where scientists sometimes get fooled by their models to think that they're studying one thing, but they're really studying something else. And I have colleagues at Duke University, and Mark Caron's lab, who's a famous scientist, and he's made just outstanding contributions to our understanding of the role of this molecule, dopamine, in human biology. And there's quite a significant body of work that says that molecule and its control is altered in kids with attention deficit hyperactivity disorder. Large body of work, and I'm not going to go through that. But following that line of reasoning, Mark studied a gene called the dopamine transporter. And it's not important what that gene does right now. Let's just call it DAT. And the DAT gene had been implicated in this disorder through human genetic studies. And so what Mark did was go to a mouse and disrupt this gene and see what happened. And I think he was thinking, like many of us, that this would have interesting behavioral consequences, but he didn't quite know what it would be good for, because there's a role for dopamine in schizophrenia, there's a role for dopamine in bipolar disorder. So what is it that the mouse is going to tell us when we disrupt this gene? And so he did that, and I think I can start that this way. There we go. All right, I don't think I'll have to tell you which mouse in this cage has the disrupted dopamine transporter gene. Now, we can sit and watch this for an hour, and he will continue to run around in this cage. This is a novel cage. He's just been placed in this cage just a few seconds ago, and this is his response to a novel environment. And you can see that he's profoundly hyperactive. So the research community, when they saw this, really was excited to think that, oh, maybe we have an animal model of attention deficit hyperactivity disorder. And the animals do have cognitive deficits. And so there's been a lot of use of this model. But over the last few years, we've become aware that this mouse may be a better model for something else, because in humans, what happens when you eliminate that gene is you don't get ADHD. What you get is a disorder called infantile Parkinsonism dystonia. You all know what Parkinson's disease is, and dystonia involves often rigid, contorted movements and positions of the body, nothing like ADHD. I thought I'd show you just what that is. This is a young girl with this disorder, and without help, this girl is not going to make it. And her life is beginning to be, the disorder is beginning to be understood and hopefully treated by knowing exactly where the genetic defect is, and it's in that protein, it's in that gene. So the transfer of information from the mouse to human sometimes is imperfect, but we learn things as time goes on, right? Now here's another, I hope you can see this. This is something we did in our lab, and this is to make another point, which is that sometimes by studying the simplest of organisms, we get clues to disorders that we weren't predicting. So that same gene I just showed you that causes this Parkinson dystonia in a young person, right, a problem with movement, we found it happening in a worm, a little millimeter-sized worm that we study in our lab, in one little corner of our lab. A very aggressive, excited group of people. Most people study mice, but some study this worm. And we eliminated that same gene in worms, all right? And now let me show you. I hope you can see. Can you see that these animals are moving? They're in water, and they don't like to be in water, and they are swimming like crazy to get out of it. Watch what happens down here. And you see these animals are becoming paralyzed. They're essentially becoming Parkinsonian with the loss of this gene. So surprisingly, not eliminating this gene in a mouse gives us some indications that we have an animal model for the disorder, but look at what happens when we do it in a worm. So sometimes this is serendipity, but it means that scientists learn from their research and occasionally get the right molecules and occasionally get the right animal model and can begin to learn something important. Now back to the mouse model of attention deficit hyperactivity disorder. We have actually worked on this. I put on a doctor's white coat. We rented a doctor's office. I enrolled subjects with the help of a true physician. I just came over to play for a day and teach the nurse how to extract DNA from subject. We collected a large group of kids who had a diagnosis of ADHD already. We brought them in, re-diagnosed them, took their saliva, extracted DNA, and we found a genetic variant in 2 kids that we thought might be causal in their disorder. And so we took that genetic change and we put it into a mouse. We made a mouse called a transgenic mouse that's now expressing that particular change. Now, it wasn't an elimination of the gene like you saw with the mouse. This was just a small, subtle change in its structure. And what happened was we had the animals and we put them in the cage just like you saw. They didn't run around like crazy, right? They have the gene. The mouse brain does not interpret that particular change the way a human does in terms of hyperactivity, spontaneous, overt hyperactivity. But what you do get, and I hope you can see this, on the top, you're going to see a mouse that's being approached by the investigator's hand. And as he reaches, you see the mouse on the top is quite docile. It doesn't really go very far. Sometimes doesn't go at all. The guy on the bottom, when you grab him, he darts, right? If you happen to read the Reporter, Vanderbilt Reporter, last week, there's a story about this animal model. And this animal model has other traits that remind us of ADHD. For example, its response to psychostimulant medications is more like humans with the mutation, like kids with ADHD. So we continue to study these animals to learn what is different about their brain that causes them to dart. But this darting response was something that was unexpected. And again, you know, what is the homologue of darting in a kid with ADHD? You know, is it that when challenged with something in their environment, a task, they become impulsive and move a lot in some way, or do not follow normal cues? These animals get picked up all the time when their cages are changed, which I think makes these guys fairly docile. But these have not yet, they can't internalize that information and use it. That's our interpretation, and we have to follow that up. And so maybe by doing the same experiment that was done at Duke, but just doing it slightly differently, we may have found a better animal model for this disorder. And we just published this about 2 months ago. All right, so now let me turn, with that as a preamble, I hope that you can see that I think that the use of animal models, yes? Yes. I think that that could be a major component of this. We are still in the early days of sampling just that. There are many ways to apply some stress to the animal and watch their behavior. You can make them swim a little bit. That will stress them out. You can play loud noises. You can vibrate the cage. All of those things are stressful. And then you can monitor changes in behavior or even their cognitive ability. And so those are early days for us. We haven't seen overt anxiety. There are some ways that maybe Dr. Emerson talked about where you can ask how often does the animal want to go out in a brightly lit area. And mice do not generally want to do that. But if you give them Valium, diazepam, an anxiolytic, they will venture out into the open spaces. So we can use that kind of test to ask, are they more anxious than a normal mouse? And they're not on that test. Thanks for the question. All right, so let's turn to serotonin and autism. That's the main topic. I just wanted to give you a sense from our understanding of animal models and psychiatric disorders that there is promise in using them, but you have to be careful and you have to be sometimes lucky. All right, so we'll talk now about serotonin and the serotonin transporter, and I'll do this in a little bit of a historical perspective because I've been working in this field for some time, and some of the contributions we've made have also been on a windy path. So let me introduce the molecule itself. Serotonin here is a very small molecule that's chemically no bigger really than the active ingredient in aspirin. It is synthesized from the simple amino acid tryptophan. We all hear about, you know, eating, Thanksgiving's coming up. Here we go, right? So we're going to go and we're going to eat all that turkey. We're going to get all that tryptophan, and it's going to make us sleepy but feel good. And does that have something to do with serotonin in the brain? Not much. But if it makes you feel good, go ahead. And, you know, you can eat a lot of things and it'll make you feel good. So, but serotonin is a very important molecule in our brain. It's actually produced by a very small group of cells, neurons, that lie down in what's called the midbrain or in the brain stem. The ones in the brain stem project down into the spinal cord and control how pain signals come up into our brain. The ones that are in the midbrain area send nerve fibers up widely throughout the brain, throughout the cortex and through memory centers and emotion centers. And there is where serotonin has its actions that we all come to hear about in terms of mood and mood disorders. So these are areas, these are called raphe nuclei. I will use that term in just a minute. So all that means is that that's a cluster of neurons in the midbrain that make serotonin, okay? Now we're going to zoom in on a synapse so that I can frame the story in terms of the key protein for this talk, the serotonin transporter. All right, here's the sending in of a nerve fiber, the axon and its terminal. And in these little circles here, we have molecules of serotonin that are being released into the gap between the sending neuron and the receiving neuron. And that serotonin is going to act on proteins called serotonin receptors. I think Ron Emerson talked about serotonin receptors last time. But these are proteins that when they bind serotonin, change the physiology of the receiving cell. Now, serotonin is sprayed out, and in a somewhat uncontrolled fashion. It's controlled by electrical impulses, but once it's out there, it's free to float around and go anywhere it wants to. And that may not be such a good thing. We might want to tightly control that flow of serotonin, and we do. And we do that with a protein that's here called the serotonin transporter, or SERT. I will abbreviate it as SERT. Okay? Serotonin transporter is a little nano vacuum cleaner. That's really what it is. It binds serotonin, it sucks it back into this terminal, and does two things. One, it hands off serotonin back to the sending side so it can use it again. And then secondly, it keeps these receptors from receiving too much serotonin, so it limits the signal. Now, we know as pharmacologists about SERT and the serotonin transporter. We've known about it for 50 years because of that power of clearing serotonin, here it is in this green protein here, back into the nerve terminal. But as well because pharmacologists have taken advantage of this powerful activity to make drugs that can block it, like Prozac. And when you block SERT, you elevate serotonin in the synapse, in between neurons, and we elevate that serotonin signal. And for some individuals, that's helpful in the treatment of their depression. Unfortunately, not near enough people. We still have miles to go before we sleep on this one. It's complicated. Depression is as complicated as any disorder. But for some, it is effective. And so this molecule, Prozac, binding to this protein, SERT, was known. In fact, the drug was developed without an understanding of what SERT really is at a protein level, or where the gene is in your chromosomes that encodes it. And that's where we came in many years ago now and cloned this gene. So this laboratory here at Vanderbilt, actually I was at Emory, and then moved to Vanderbilt, identified the gene that encodes this Prozac target, SERT. Okay? And that sort of got my lab off and running. One of the exciting things about it, I'm not going to go through the data in this paper, but one of the things that was most exciting to us when we identified this gene was finding where it is on your chromosomes. So we thought, and here it is, we located it on chromosome 17. This is just what's called an ideogram of a chromosome, and these little dots indicate our confidence in knowing exactly where it is. This was pre-genome project. We didn't have the sequence of chromosomes. We couldn't just look it up on the web and find it. We had to go hunt for it in billions of bases of DNA and found it on chromosome 17. And when we did that, we were extremely excited that we might break through an understanding of mood disorders, depression, and suicide. My family is racked with that, and this was very exciting to me that I might have my contribution in science through this work. And as it turns out, we made some other observations, and it's pointed us in a slightly different direction, a big different direction as we've moved toward autism. So I'll tell you that story now. It begins with this finding. This is a slice through a mouse embryonic head. Okay, so I'm looking now at a mouse head in the middle of gestation, so embryonic day 13. You can see the eye here and the mouth. And what we're doing now with this section is applying a probe to it to identify where the SERT gene is being made. And what we found, to our surprise, was that in this embryo, there is a lot of this gene being expressed already. This is not an animal yet that's experienced the world, experienced its interactions with others. Nothing that we could think about in terms of mood disorders would come to anybody by looking at this. And we began to think that maybe there's a role for this protein during development, during brain development. And others have gone on to do very elegant studies to show that if they manipulated serotonin just the right way, the brain doesn't develop properly. And there's changes in sensory function. There are changes in later life anxiety, and we began to think that maybe the study of this gene should focus on disorders that emerge during early development as opposed to disorders that we typically associate with adolescence and adulthood. So that was our first finding and caused us to think about moving into a different disorder. Here's another finding by another group. Again, it's just, these are sections of the brain, and it doesn't really matter that you know anything about anatomy, just that you can tell the difference between these dark staining patches and the areas that are not. And what we're looking at here on the top row are sections through a mouse brain at embryonic day E14.5. So the animal, a mouse's gestation is 21 days long. So this is the 14th day of that gestation. So about to go into the third trimester, essentially. And you can see that there's already dark deposits of this material, which is a probe for those receptor proteins that bind serotonin. So they're already heavily expressed. You see one type of serotonin receptor here and another here. You go a few more days in development, you get more, and you get more. So the brain is set up to respond to serotonin as embryos. Okay, that's a really critical and exciting finding that occurred not very long ago. So this is a paper published in 2006. Okay, so we've just begun to learn about the role of serotonin in brain development. Another finding that we published with our colleagues in our Conti Center here, Pat Levitt's group, who's now at USC, his fellow Alex Bonin discovered that the placenta is actually a source for serotonin for the developing brain. And so this occurs in mice, and it occurs in humans as well. The placenta makes serotonin and ships it to the developing brain, and that serotonin is important in establishing the appropriate architecture of the brain. Later, that raphe, here's the dorsal raphe, remember those raphe neurons in the midbrain? They take over, and they begin to supply serotonin. But during embryonic development, the placenta is actually participating in this role of organizing how the brain works using this molecule serotonin. Okay, well that then sets up for me thinking very strongly we need to be thinking about serotonin in developmental disorders. And there are some other aspects of this, and I'll come to it in a second, that kept us on the trail of serotonin in autism. But let me remind you about some of the statistics associated with the disorder. The CDC has reported in 2014 that 1 in 42 boys could be diagnosed on the autism spectrum, 1 in 189 girls, so a ratio of 3 to 4 times more prevalent in boys. And we still don't understand that. The sibling risk is 25 to 50-fold above the population risk, suggesting that there is something genetic and common running within families that can be shared and driving risk for autism. That's a really important finding, because that means we might have some luck in finding clues by studying DNA. And there's a 10-fold higher risk for a subject who is a monozygotic, meaning they're identical twins, as opposed to fraternal twins, again suggesting that genetic heritage and genetic identity play an important role. The disorder itself of autism, and now we think about it more as a spectrum, autism spectrum disorder, is made up of a cluster of differences in behavior. So one, we have difficulties with social interactions and social reciprocity and inferring meaning from another. There are language deficits throughout development that can be very severe and can really hamper intellectual development. And then there are rigid compulsive traits, and they appear in many ways. Rigid attention to certain kinds of objects, only wanting to eat certain kinds of foods, rapidity and repetition of certain movements, hand flapping, for example. So this is a constellation of symptoms that are obviously complicated in and of themselves. And to get a classical diagnosis of autism one need to be right in the middle and have all three of these things. We now recognize that there are relationships that occur among individuals who don't have all three, but maybe have two or even one of these. You've heard of something like Asperger's syndrome, which the term is moving a bit out of vogue simply because we like to think about the spectrum of autism and not try to categorize individuals particularly. But those individuals typically associated with higher functioning individuals, Asperger's syndrome, and you don't see these language deficits. You will see problems with social interactions and sometimes some other behavioral traits as well. So anyway, these three domains are the areas we think about as we think about how would an animal model of autism look, right? Could we imagine having an animal that we could tell had language deficits? How would we see that? How would we? I'm not Dr. Doolittle, and so I'm Dr. Blakely. So I'm not sure that this one is one I can tackle well. I'll show you. We've tried just a little bit. Rigid compulsive traits. How many of you ever had a dog that won't stop chewing his foot, or turns and spins in circles, or has his favorite toy and just won't let it go, and that's it? So maybe we can get close there. Social interactions for sure. Animals, all animals, at some point in their life have a social interaction. If it's nothing but with the mother and then they go off and lead a solitary life. But usually they do get back together and reproduce, so we've got that opportunity as well. But many, many animals have rich social lives, and so we can tap there. So I mentioned the genetics of autism is strong. It's strong, but it's complicated. This is an set of chromosomal illustrations all the way through. You can see each one of them. And in these red dots and green dots represent areas that scientists have found a reason to implicate that region or a gene in that particular region with risk for autism. This is staggering, the complexity of what we're dealing with here. There's not a gene for autism. There are genes for autism risk. And that's that last part is really important. That risk is not a sentence. It just is a probability that if the rest of the stars align, and some of those stars could be environment, they don't have to all be genetic, then the outcome could come with the autism spectrum disorder. But we also believe that even though it seems daunting, that there are secrets in the relationships of those genes. They're not all functioning independently, right? We know we have genes that influence the way our body develops, and they work in cascades and they work together. And very likely many of these genes work together, and if you insult one of them, you can have the same outcome as insulting its partner gene. But it is complicated. So we need other clues. And here we come back to serotonin. Is following the trail of serotonin relevant for autism? Well, more than 50 years ago now, a group led by Danny Friedman showed that kids, individuals with autism, have an unusual blood chemistry in that they have elevated serotonin in their blood, a significant fraction of them. It's about 25 to 30%. And the way this looks is shown here in a nice study in 2004. We have a distribution of serotonin levels in the blood from low to high. And what you can see, I hope, is that this is the normal range. And this is from a group with autism, and they're all normal. But there's this other group out here with high levels of serotonin. This tends to be predominant more in the males, and again, this is a male predominant disorder. So this has been a clue, and you don't see this in other disorders and other individuals. They all sit over here. So what is this elevated serotonin about? What is it telling us? Is there something about the biology of autism? Well, we thought so, and we thought so not just because of that finding, but because of some others that relate serotonin to the brain. And just let me review a few of those with you. So first, where's the serotonin in the blood coming from? you may be surprised to learn that it's actually coming from your gut. So in the lining of your gut, there are endocrine cells. They're called enterochromaffin cells. They make actually 99% of the serotonin in your body. There's very little in your brain. It's very powerful, but there's a lot more that controls your gastrointestinal function, and it spills over into the bloodstream, and it's picked up by platelets that you know are involved in clotting, the clotting response. And it turns out that they use serotonin in the clotting response. So there's a completely different side of serotonin biology that exists in the periphery. Now, the important thing is to make serotonin, to package it for release, and for the talk today, to suck it back up inside cells. Platelets use the same gene, same protein that your brain does. So maybe our thought was, if we're seeing a signature in the blood, could it mean that there's something about this gene and this protein that's also altered in the brain, in the same individuals? And how are we going to find that out? Because I can't take the brain out of those people. I have to be smarter at it and work a little harder. So let me tell you a few other things, though, about the serotonin. So there have been reports that in a platelet, here's a diagram of a platelet, and here's that serotonin transporter protein sucking up serotonin so that the platelet can use it and re-release it at the beginning of a clot. So there have been reports that this protein's activity or levels are elevated in autism spectrum disorders. There are brain imaging approaches where we can actually look at how serotonin synthesis develops over time, and individuals with autism show an abnormal pattern of development of their synthesis capacity. And lastly, if we take away tryptophan from the diet and deprive these individuals of the ability to make serotonin, this is difficult to do. You have to, it's a very controlled thing. It's not just like, well, let's go on a tryptophan-free diet by going down to GNC and getting some supplements or something. That's not how it's done. It's very difficult, actually. But when it's done correctly, what you find is that this worsens the symptoms of individuals with autism. And so we think, therefore, that by inference, serotonin has something to do, or maybe the lack of it, with this disorder. All right, so our physical entry into this field, however, came with a collaboration with a geneticist here at Vanderbilt, Jim Sutcliffe. And what Jim was doing was trying to figure out where, just like those diagrams I showed you, where on particular chromosomes are there hotspots for risk for autism. And here is a plot of that kind of risk. And this is from a group of families, 327 families with autism. They have multiple individuals with autism in the family, and they also have to have an unaffected child. And the game is, what part of the paternal and maternal DNA was given to the kids with autism that was not given to the kids that are free of autism? And when you perform that study, what you find is that there's an area on chromosome 17 that is a very, very strong peak of risk. This is in black, all the families. If you take out the families, so 138 families, if you take them out of the analysis and look at them separately that have no males or that have at least one female, and they might have a male but they've got at least one female, you see that the risk collapses in this area, right? So there's something about male predominance in autism, we think, that involves this area too, and you see that peak goes up. These are the families with only affected males, okay? Very strong signal. And that, of course, was exciting to me because if you remember back when we cloned the serotonin transporter gene and thought we were on our path to studying depression, it was on chromosome 17. In fact, this is where it is, all right? So this is exciting. There's a little bit of, I'm using a little license here because SERT is here, but it's surrounded by hundreds of other genes. This is a big area. So we weren't really sure that there's something different about SERT in some of these individuals, so we just decided to go and look. And so we took the gene apart, and here's what we found. This is a diagram of the serotonin transporter gene, and you can see that there are these little vertical dashes here. They represent pieces of that gene that are stitched together to ultimately make the protein. And in yellow, what you see are changes in that gene that affect the protein, that make a different serotonin transporter protein in an individual with autism. We screened 24 subjects, and we found, actually there are 5 here. So 5 changes in that gene were identified. So 5 out of, we actually got 5 out of 100, but we got 4 out of the first 24, right? Now, the frequency, if I sample this room and I ask how many people have a difference in their serotonin transporter protein here, there's probably 1 change in 100. That's what the estimates are. Well, we took 24 of you and found 4. And that, to us, was improbable, statistically improbable. So we identified those, and 3 of them had never been seen by anybody else before, and still haven't been seen. They're rare. They're within individual families, but we thought they might confer the risk. What were they conferring risk to? And remember, it's a big spectrum. Autism's a big spectrum. You know, lots of different, the clinicians will say if you've seen one kid with autism, you've seen one kid with autism. So it makes it very challenging to treat, but it also makes it very challenging to figure out, if I have a genetic change, what's similar about that one that's to another individual with a different genetic change. So we have to pile up a lot of subjects, and we had them. And what we found was that where they were most associated with changes in this one gene were in these rigid compulsive traits and another component of autism that's often seen called sensory aversion. Sensory aversion means having a strong reaction. Can be negative, can be positive, but most often negative, to being touched or to sound. You may see kids with autism with cotton in their ears or headphones. They're very, very sensitive to touch. And so these 2 particular trait domains we have our eyes on. If we're making an animal model and it involves the genetic material I've just showed you, perhaps this is where we will find evidence that we've actually accomplished our goal. Now, I need to introduce you just briefly to the actual protein I'm going to talk about, and I've been talking about, the serotonin transporter. This is what it looks like to X-ray crystallographers. It's this coiled up set of ribbons and helices that ultimately bring the molecule serotonin right through the middle. It's just like a big vacuum, right, where you're pulling serotonin from one side to the other. This is a cartoon representation of this protein. It snakes through the membrane. This is the cell surface. Here's the outer world. Here's inside the cell. So it's going to be bringing serotonin this way, in from out to in. And it's got these 12 barrels that fold together to make this. And we could find where these changes were in the protein and begin to study them. We could ask questions like, are those parts of the protein important for things that we already know in terms of the protein's activity or regulation? Are they at spots that across evolution don't vary? And that's the signature that that's a really important site. Right? Over millions of years, there has not been a change in an amino acid at that place. And there are other features of protein biology that finding where they're located is important. I'm going to focus on this one here because this is in this domain that sits inside the cell, because work in our lab and in other labs had showed that that particular region is a master switch for controlling whether this protein is working or not working. And it turns out that just like a switch on your vacuum cleaner, neurons can do the same thing. They can turn it off and they can turn it on. They can turn it up and they can turn it down. And that's important for the fine-tuning and the control of the use of serotonin. And we had mapped it into this area. So when we saw this particular change here, we were very excited. And I'll tell you some other features of it before we get to an animal. Let me just show you the pattern of conservation of these changes. So this is one of the variants here. I've got a lineup of the amino acid sequence written in single letter code. So each letter is a different amino acid. And you see human, cow, chicken, fly. You go all the way down. Then there's some dopamine transporter proteins, DATs, and norepinephrine transporter nets. They're sister proteins. And we can line up their sequence too. And what you can see is that one of these variants hits an amino acid that's conserved all the way from human down to flies. Okay? So that's an important sign. That's an important residue that you do not play with. Messing with that could cause problems. Here's the family in which this variant was transmitted, so you can see how excited we were about it. It's carried by the mother. The open circle aspect of this means she's unaffected. She transmits it to two boys, filled squares. Boys are squares. We know that. And they're filled, meaning they have the disorder, right? So the two boys who get it have the disorder. The boy that doesn't get it doesn't have the disorder. All right? She also transmits it to three female girls. They don't have it. They don't have the disorder. And then there's another girl that didn't get it, and she doesn't have it either. So this has that pattern of transmission of a risk gene, exactly what we're looking for. Selective transmission to the affected subjects, and it's to boys, right? Because remember where that gene was located, that was a risk area for boys with autism. So that was a very exciting finding. Here are two other places in this protein, same kind of alignment, that show you that these positions are even more conserved. They're conserved even across the sister proteins. So again, this isn't something that should change without affecting the function of this protein. Now lastly, let me show you the amino acid that I showed you before. This is the one we're going to focus on. It sits right here, and you can see it's conserved human, mouse, rat, cow. You get to chickens, and it becomes the substitution that we actually found in the human. It becomes an alanine, or A, from a glycine, or G. So this alanine at position 56 is the substitution we're talking about. And I had some astute person ask me if the reason that we found it was because the subjects had more chicken-like features. And I thought, no, you know, come on, this is a very different protein. Of course, it's changing. Then I had to stop and think for a minute about what the genome was that had been sequenced that was the chicken genome. And I don't know this for sure, but my guess is it might be one of these highly inbred chickens that has probably extraordinary repetitive traits. And so maybe the person wasn't off so much. Maybe they're offhanded, and maybe even callous sounding comment has a tiny little grain of truth in it. So I'll tell you what happens when you switch this to an alanine in an animal. A couple of other features. Excuse me. So about 3 million Americans carry this, right? This is carried by about 1% of the U.S. population. It's not within one family. This is the most common of the variants we found. This is why we went after this one, right? We felt like we could ultimately do human studies and get enough individuals to study them. It was 4 times more prevalent in our subjects in that study than over the population. It was over-transmitted to autistic subjects. And if you were a male and you had the variant compared to a female had the variant, you're more likely to be autistic. So these features told us, study this deeply, and we did. And we have, and we do. This is a major activity in our lab right now. All right, now, this is a plot that shows us how active the serotonin transporter is. SERT means human SERT. All of these are human forms of the serotonin transporter, and the length of the bar indicates how much activity it has. Serotonin is abbreviated 5-HT. So what you can see is that all of these mutants, including the one that I'm focusing on, have excess activity. That might be a surprise to you, was a surprise to me. I think I, like many people, tend to think that we change the protein and it becomes a worse protein. It becomes non-functional or doesn't work as well. These actually, under these conditions, work better. They're like, they're in hyperdrive. And the idea we have now is that they're taking serotonin away too fast and at too active a rate. That's depriving the brain of the serotonin it needs. And remember what I told you when you take autistic subjects and you take their tryptophan away, that their symptoms get worse. Is that a clue that we're seeing the same thing with these hyperactive proteins? Well, there's another feature of these molecules, and it's shown in this graph. Now, when we did those other studies, we took the serotonin transporter gene and we put it in cells that don't have it, and we assayed how much transport function now arises in those cells. So that's what those plots were. That's a very artificial situation. And so we wanted something from the individuals. Well, I'm not going to get their brain. I could get their platelets, right, because it has SERT in it. But these subjects came from all over the world. So the chance for me to go to Finland and get blood from subjects, and some of these are not even known to me. I don't even know their identity. I just had their DNA. But I had more than their DNA. I had their lymphoblasts, or their lymphocytes. That's how investigators actually bank DNA, because they collect the individual's lymphocytes and they immortalize them so that they will grow forever in culture, and then they can freeze them down and bring them back out later, make some DNA, and give to an investigator, and give to another investigator. So it's a bank of DNA. Well, we realized that the lymphocytes, too, this is a discovery that had happened just a few years earlier. Lymphocytes express SERT. So we said, go get the lymphocytes that are in the bank, and let's pull them out and see what are they different. And they were. They were all hyperactive, just like I said. There was another thing we found with them that was even more striking. And that is, remember I told you that SERT uses that area to turn the activity up and down? And we've discovered how that works. And we can make it happen with simple chemical manipulations. And we've done that here. I've used a compound to stimulate a protein called PKG. And what you can see is that SERT activity jumps up. You get more activity. And you can use another drug that targets this protein, and it also increases. That's in a normal SERT. As we look at the copies of this mutant that are introduced, and we get to individuals who have 2 copies of this mutant protein, you see they don't increase at all. And these have the most elevated SERT activity. They have 2 bad copies. These have 1, there's a step down, and then here there's no stimulation whatsoever. The interpretation for this, and it's a simple one, but it's one I think is correct, these transporters are in the on state. It's the gas pedal is down. The vacuum cleaner is on high. And there's nothing you can do to it. You can't move it up. It's already there. And that's what's happening with these guys. Okay? So this was a really important finding because it also told us about some other things that could be changed that make this protein be hyperactive. And it's those other things that may be clues to other genes that are participating. Mm-hmm.
The one on the top, somebody on the autism spectrum,
Yes.
Not
Not really. They have the disorder for sure, but many here as well, and some of these are severe as well. There's so many other factors that modify how it expresses itself, but at a cellular level, you can really see the differences. All right, so we've gone through a bit about serotonin and a bit about serotonin autism, and now let me spend the rest of the time here telling you about this mouse model of autism. So we have made a mouse, engineered in it that particular variant, that ALA56 variant, We put it into the mouse genome instead of its normal glycine, and what happens? So first off, what you see is that the animals look normal. Don't forget the coat color, that doesn't matter in this case. It's just the size and the reproductive fitness and their growth and all that. That's all normal. But when we sampled the blood, look what we found. These animals are hyperserotonemic. They have what we call a biomarker. They have this mysterious elevation in serotonin that we didn't know how it could come about. And we just showed now for the first time that we can make that happen by messing with this one protein. And we think it's because there's too much serotonin being sucked into platelets. And the platelets fill up the bloodstream. They're the major source of serotonin in your blood. I'm going to skip this for time. Okay, so we made these mice. What happens with their brain? So there's a stain that you can use to look at how those neuronal projections, those axons, are going from neurons and innervating the brain. And you can monitor their trajectory. They're shown here in green and in here in black and white. Let's just look at the black and white. What you can see that in a normal animal, there's a really tight band of these nerve fibers moving along through the cortex during development. In the ALA56 animals, you see that it's spread out. It's much wider. These axons are wandering. They're not developing normally. The brain is there. Grossly, it looks normal. But the fine patterning within the brain is altered for these nerve fibers. Now, what you really want to know, I think, and this is what I wanted to know too, is how's their behavior changed? What do they really look like? What features of autism might we see in these animals? So to tackle that, we've turned to the Laboratory for Neurobehavior. This is a core facility at Vanderbilt. It's a state-of-the-art, one-of-a-kind facility. It's the whole floor of MRB3's 9th floor. And it's segregated into many testing rooms. You heard about that if you came to Dr. Emerson's lecture. And in each one of those testing rooms, we have automated, video-based data collection instrumentation to monitor many, many things that these animals do. And I'm just going to talk about a few of them. There's a few of the rooms with their instrumentation, surgical suites, and we can study learning and memory, we can study emotionality, we can study anxiety. Many different features of behavior can be dissected. Can you visit? It's a controlled access facility. The animals have to be kept under quiet condition, and there are tours that happen externally to that facility that are like, I could give you a whole hour on aspects of it, and so it maybe on another one of these meetings, it would be good to have the director of the facility come and talk more about that. So remember those domains of deficits I talked about, and I made the joke that I wouldn't know anything about language. So one thing we can do is we can monitor the squeaks that mice make. We don't understand what they're saying. We can put them in situations where squeaking can probably be interpreted as a call of distress or a, the love song of, J. Alfred Prufrock or something, I mean, when we got mating, I don't know. But in this particular case, what we're doing is we separate the pups from the mother, and then we get ultrasonic vocalizations, things you can't hear, but you can hear them with the instrumentation we hear, as these pups chirp to get back to the mother. And what you find is that the individuals, the mice, the mouse pups, and there are some normals in here as well as some of these mutants, the ones that carry that ALA56 variant don't chirp. Somehow they're impervious to this particular manipulation. Now, it's not that they can't chirp, they just have no interest in it. As far as we can tell, they don't emit it. And so that's significantly reduced, and maybe that has something to, maybe that is either a social thing or it could be some kind of communication deficit that we're seeing. All right, what about that repetitive behavior domain? Remember that one I talked about, that individuals with autism have specific fixations or repetitive behaviors. One way we can assess that is, of course, just to watch them. But when humans watch animals, animals freak out, right? They know we're watching them. So what we do is we put them in isolation chambers, right? They're perfectly healthy, they've got a happy environment, and there's a video camera watching them, and a computer is decomposing all that video and telling us what they're doing. So without human intervention, we can calculate how much time they spend eating, rearing, grooming, hanging, lurching low, which is the behavior that mice do, sniffing, stretching, whatever. And this is only a small group of hundreds of things that the computer will tell us. And what you can see is that there's one thing that's different in all of this, and it's called hanging. What is hanging? That's what I wanted to know too when a student came back and showed this hanging thing. So what it is that it's the animal hanging at the top of his cage. Attaches a little wire mesh on it, and he goes up and hangs on it. Or he goes up and touches it. It doesn't have to actually physically hang. What we're showing is the time spent hanging. Well, the time spent hanging could mean several things, right? It could mean that the animal goes up and just hangs out, right? Or it could mean that the animal goes up and back and up and back. And I think you know where I'm leading you, right? That repetitive behavior of going up and back is what these animals do. And here we can determine that by just plotting the number of times they execute a hanging bout. And what you see is that these animals that carry this mutation exhibit 1,000 bouts of hanging in a 24-hour period. Remember, they're sleeping 12 hours of that. So this is an extraordinary repetitive behavior they're engaging in. And it's not something I could have written to the NIH and said, I am going to study hanging behavior in my mice. It's something that emerges. And this is one of the things that scientists understand when they review proposals of their colleagues, that we can't predict everything. We have to monitor, we have to watch. And this is one of the things that we found. Okay, here's another test we can do. This is now in the social behavior dimension. This is called the 3-chamber test. There's a plexiglass partition here. A mouse is put in the middle, and there are little doors, little openings that it can go to either side. And it goes to either side depending on how interested it is in whatever is in a little upside-down pencil cup here that can't go anywhere. It's a little cage. You can see it. So you can put a familiar mouse or a novel mouse, and the mouse more often will go over and check out the new mouse, right? Or you can put an object and a mouse. And the mouse, as shown here, a normal mouse will go over and stay with the mouse, not with the ball or the little stick thing. These mice don't have social preference. They don't care whether it's a mouse or a ball, right? So this is interesting because this is a characteristic that it's a social dimension of behavior that seems to be affected in these animals, and it may be something akin to what individuals have with autism, although we don't quite know. Yes? Did they say it one more time? Will they prefer a male versus a female? No, not so far. Not so far. I mean, they have this preference, this differential preference between objects and mice, but the differential preference between different mice is not something we've picked up. All right, but we have picked up an encounter phenotype. It's another social phenotype, and it's really important that I tell you about this because it leads to an exciting potential breakthrough therapeutically, because it's such a simple test to execute. And that is called the tube test. And in this test, Mickey here is asked to run down this tube. You train it very quickly, a little poke in the butt with a pencil. The mouse knows the next time I come to this tube, he's going to poke me again, and I'm just going to run down this tube. Well, you fool him one day, and you put the other mouse over here. My colleague calls this the McDonald's Playland test. And here comes another one coming up the other way, and they meet in the middle. And all you need to do here is score who backs out. They call this in social behavior circles, if you are the one that pushes the other mouse out, you're the winner. and you're the loser if you back out. All right? But don't take this in a pejorative way. It just means that you're getting away from this social encounter. And what you can see is that these mice are not the winners. They're the losers. They preferentially back out of this encounter. I'm going to skip the gut stuff. All right. So those are the phenotypes. Those are the major things we've seen in these mice, and we're very excited. We have engineered with a very subtle change. It's actually one base of DNA has been changed. And these behaviors have become obvious to us. We now need to know where in the brain and the body is this variation changing behavior. This SERT protein I told you is in platelets. It's in lymphocytes. It's actually in the placenta too. It's in the brain. It's in the lung. So it's in many, many places and probably having a pervasive effect on lots of different processes. Which of those are the most important ones for the development of some of these behavior traits? We need to figure that out. How is this little subtle change in this protein changing its activity so dramatically that it's changing behavior? We want to know what other genes might be controlling a normal SERT but themselves be defective. We could get to the same endpoint. And maybe that's a clue to that network. And lastly, could we use this model to search for new treatments? Did I hear? Yes. Yes. So the question is I think, just is whether or not this is an aggressive encounter between the one animal and the other. And certainly there can be vocal emissions happening here. The animals can't pass each other, they can't attack each other, but there can definitely be fear on the part of the animal that's got the ALA56. It could be sensory aversion, doesn't want to touch the animal. There are a lot of interpretations, and they have to be teased apart by other kinds of tests, just as you're suggesting, looking at complex social interactions, looking at cases where you introduce an aggressor mouse and ask if this animal is one that will cower more than another. Those are ongoing tests right now, but it's a good point. Okay, just let me leave you then with this aspect of therapeutics. So one of those pathways that I showed you that can turn the transporter on, I showed you a graph of that, just the normal transporter, has been under intensive study in my lab for years, going quite independently of the finding of these mutations. And we identified a molecule that can actually block that stimulation, a drug that can block it. So if our transporter has too much activity, maybe we can bring it down if we use that drug. And so our colleague at Northwestern, Marty Watterson, has developed a drug and sent it to us, and we've started putting it into our assays. Let me just show you one very promising test. Again, it's the tube test. What I showed you before, and we've repeated this many times, is that these animals don't win if they're given, in this case, saline. But look what happens when we give these animals this drug. It's called MW108. Now they're just as good a winner as the other animal. They carry out this social test normally. They also, this drug also normalizes several other physiological properties that we've seen to be disrupted in these animals I didn't have time to go through. So we're very excited that actually having built this animal model is going to allow us to have some kind of testing ground for new drugs, new therapies. Because without it, I would be forced to say, let me take a normal animal and give it a drug and see what happens. And this is a slippery slope in our development of drugs. It's a real problem. And in fact, maybe why many drug companies are getting out of the development of drugs for psychiatric disorders is because they've been relying on normal animals. And we know that normal individuals respond differently than individuals who have a problem. a drug that might help an individual with a problem might actually even have negative effects or no effects on a normal person. So we're excited. We've built an animal model that has some features that do remind us of some aspects of autism. Our main excitement really is that we've been able to connect that gene, the serotonin transporter, and that protein to a suite of behaviors that can be influenced by human genetic variation. And so we'll see how it goes. We'll hope you'll follow our work in the coming years and see whether or not this has been a fruitful path to take. So I'm going to stop there, and happy to take your questions. Here we go. Yeah, the question is there value in getting your DNA analyzed? There's several answers to this. I think that to some people, they don't want to know certain things. They want to let life play itself out. I'm one of those people, particularly when there's risk for something that impacts the lives so severely of children and families. You want to know everything you can find out so that you can treat it better and that you can help diagnose. So the question is whether someone who doesn't have a particular disorder still wants to have their DNA analyzed. I have an uncle who's about 95, and I've had my DNA analyzed, and in my excitement I said, you know, Bob, you should get your DNA. This is really cool. You'll learn a lot about yourself. And he said, it's too late. And I didn't mean it that he was just going to go read his DNA and he was, you know, his life was going to turn around or something. I just was just an innate curiosity factor. So I think if there's a lot to learn about yourself, very revealing, of course, your heritage, where your origins are. I've had, by DNA analysis, I have had relatives that have had pictures of my family from the 1800s contact me and give me those pictures. And it's just remarkable what we can do with that. Have I learned something about my medical history yet, or what my medical history may be? Not really. There's a lot that we don't know. So most of the tests out there, most of the things out there, if they're very targeted risk factors in your family, and you want to know whether or not they might be transmitted to your children, then those things perhaps should be looked at. But for the most part, a lot of the DNA companies that are out there selling kits to look at your genome, it's of questionable value in my mind. We're still not there. We will be one day. I think we'll understand a lot more, but so early in the game that I don't know. I don't know what you learn.
You talked about chemical findings. What about neurodegenerative change?
Mm-hmm.
You know, at autopsy, using electron microscopy.
Right. Right. This is so the question is, I think, have people looked at the brains of individuals with autism who have died and gone in and looked at pathology? There's surprisingly little information on this. You know, in order to make comparisons, you need a lot of subjects, and you need to have known circumstances under which they died. There are medical issues that arise with individuals with autism, and so some of those could cause some changes in the brain. But the total collection of brains that we have numbers in the teens. So the population's small. So we still don't have, we don't see, again, gross pathological changes, but there is some evidence of microarchitecture changes in certain cell populations in the brain, and it's gotten a number of scientists very interested and excited that there are some things happening early in development which could be seen way off in later life. Remember, one of the things about autism is that it's not just that there's a disorder, it's that you live with it for a long time. So it's not that the individuals die early and we have them dying all the time so we can get their brains. They're going to live with their autism in many cases as long as any of us. And so that's a huge burden for them and on their families. And there's a lot that's often thought about in terms of children and treating children. But the group that's really underserved are the adults with autism, in my mind. Yes. 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 Right. In the individuals I talked about, they have a change just in that transporter protein that will control how much serotonin is available to those receiving proteins, those receptor proteins. But you could get to the same point, we think, via changes in those proteins as well. And the number of those proteins is very large. It turns out that there are 15 different kinds of receptor proteins to respond to the one chemical signal, serotonin. And they're distributed in complex ways, in complex patterns. So figuring out which of those proteins or which collection of those proteins is involved is a big effort that's underway right now, but it is difficult, and we don't have big clues on that particular part yet. Which receptors? Yes?
I assume that once you create this model, it's carried down through generations as it reproduces.
That's right.
Have you found any difference between the male and female offspring of those mice?
We're just beginning to study the pattern of early development of these mice from the time in utero to as they go forward. And as a result of that, the differences in males and females as they go forward hasn't yet been analyzed, but it's on the books. We would like to know that. We'd also like to know, is there an important role of the mother's genotype, right? And I think you all know, you certainly lived through a period where mothers were blamed for their children's autism, right? Do you remember the refrigerator mother syndrome, right? This was depicted. And, you know, this just cast a pall on understanding this disorder and blaming people for something that has such high heritability. But the issue is that we all carry genetic variation. We're all contributing something genetically to our offspring. And the question of whether we contribute something there, not from poor maternal care, but maybe just from genetic changes. Are there physiological changes in the mother that the offspring, also carrying the same change, is now responding to differently? And it's actually it's a bad thing on one side and a bad thing on the other, and together this is a problem. So there are studies underway there. There are some actually exciting data on that beginning to emerge from laboratories at Vanderbilt and now also at Columbia studying these mice. So stay tuned. I think you'll hear something about that.